Hi everybody, today we're going to be talking about translation. So we already covered how DNA gets to protein. We said that DNA starts and then it gets transcribed into mRNA, and we've talked about transcription. And now we're talking about translation, where the RNA and the mRNA becomes a protein. So as always, what are the starting steps? So first we have our mRNA strand, and then we need our ribosome. In the ribosome, we have a large ribosomal subunit and then a small ribosomal subunit. Now within the subunits, there's different types of rRNA. And if you recall, we have three types of RNA. Messenger RNA, which is mRNA, ribosomal RNA for rRNA, and then the transfer RNA for tRNA. In prokaryotes, which is highlighted in green, we have 16S ribosomal RNA within our small subunit. And then in prokaryotes in the large subunit, it's 23S. In eukaryotes, however, it's 18S rRNA and then 30S rRNA. So that's just the type of rRNA we're using. Now, what does the S stand for? The S means Svedberg. That's a sedimentation unit. So whenever we take this component, if we were to spin it and then it'll sediment down, which is like little particles of grain, that's what this is measuring, and that's what that number signifies. So don't be scared when you see that 16S. So we have the 30S and the 40S in the eukaryotes for the small subunits, and then the 50S and the 60S for the large subunits. So what does that mean? We're looking at the size of the prokaryotic ribosome. So the 30S, which is a small subunit, plus the 50S equals 70S. But wait, 30 plus 50 doesn't equal 70, it equals 80. However, if we were to put these together and sediment it, it comes out to 70, and that's what that means. The size of the eukaryotic ribosome, we have 40S plus 60S equals 80S. 40 once again being the small unit, 60 being the large subunit, equaling 80. Now here's a quick trick. This is something that's tested on the MCAT a lot. So we go 3, 4, 5, 6, 7, 8, and then if it's even, E for eukaryote. So that's telling us 40 plus 60 equals 80, and then we could say 30 plus 50 equals 70. Okay, let's move on and talk about the ribosomal sites, now that we've kind of classified what the ribosome is. So we're putting sites on this ribosome. We have the A site, the P site, and the E site. The A site is the amino acid site. This is where the tRNA enters, and also this is where the mRNA first attaches and gets in. The P site is the peptidyl or peptidyl transfer site, where the tRNA anticodon loop matches with the corresponding mRNA. We'll talk more about that when we get to the specific steps. Then we have the E site, which is the exit site. This is where the tRNA leaves. The exit tunnel is also present, and then the peptide bond is formed when the amino acid has to form a bond with the next amino acid. So let's talk about the translation steps. So once again, we have the ribosome and we have the mRNA. And you can move here a little bit. So the tRNA has to be charged first. That's the first step. What does that mean? So we have 20 tRNAs that correspond each with a single amino acid. And that's why we learned the 20 amino acids. So the enzyme amino acyl tRNA synthetase that will be tested is taking our tRNA, hydrolyzing ATP, which basically means it's using the power of ATP to take it, and then the corresponding anticodon loop will correspond with the correct amino acid. So for example, we know that when we look at the mRNA strand, that methionine goes with AUG on the mRNA. So that means on the anticodon, it should be UAC. And that's what that loop is representing, and that's how the methionine is being charged. Without the amino acyl tRNA synthetase, translation would not happen. And one important thing to note is, unlike transcription where there were several differences between prokaryotes and eukaryotes, there is no differences besides the size of the ribosome between prokaryotic and eukaryotic translation. So let's move on. So here we have our charged amino acyl tRNA, and then we have the mRNA. It has entered into the A site and then the P site. So the tRNA is going into the peptidyl transfer site. It's corresponding with the AUG and the UAC, the anticodon loop, and then the methionine is knowing, okay, this is where I need to go. So how does this happen? How does it know exactly which one? It's not perfect the first try. each of the 20 tRNAs will go and try to sit on until the correct one makes that bond. So each tRNA attempts to sit in the P site, but only the correct anticodon loop can pair with the corresponding mRNA. After that, the next tRNA that's charged will come in, and the first one will go into the E site and the exit site. You can see that we have our amino acid, or specifically our methionine here, sitting in the exit tunnel. The exit tunnel is where the amino acid chain forms, and then that tRNA exits. So the next tRNA comes in and attempts to match. And the first amino acid is bonded with the second amino acid here after the match, and this is forming the peptide bond. And we can see that we have UAG, which is corresponding with AUC on our mRNA. And once again, it's 5' to 3' on the mRNA, and 3' to 5' on the anticodon loop. Quick reminder, what does 5' and 3' mean? 5' is the phosphate end, 3' is the hydroxyl end within the 3' ribose sugar. So the process continues. We have our peptide bond here, and this would keep going and going, starting with the charging all the way back to here, until a stop codon is read, which is UGA, UAA, and UAG. So you go away, you are away, you are gone. That's how you remember it. So let's talk about the wobble hypothesis, now that we've covered translation and everything that's going on. So the wobble hypothesis is stating that within that anticodon loop, the first position nucleotide may be this thing called inosine. It's a different nucleotide and it looks similar to ATGC and AUGC. And this is present there because it helps prevent mutations. So basically, if we limit one more variable that could potentially be leading to a mutation or a point mutation, this is what the inosine is helping to prevent. So the other thing about the wobble hypothesis is that whenever tRNA is trying to pair, when we're trying to get the tRNA to match, it's not a perfect match. tRNA kind of wobbles. So the role of the wobble hypothesis is to help state the tRNA wobbles when it's trying to match at the P site and it's never a perfect match. And the second thing is that inosine helps us to prevent mutations. Those are the rules. So quick recap, we talked about the size of the ribosomes, the difference between eukaryotic and prokaryotic ribosomes, then we talked about the steps of translation. Now here are some questions that could be potentially on your exam. So what happens when the exit tunnel is blocked? Well, that's actually the exact mechanism of macrolides, which are antibiotics like vancomycin and azithromycin. If you've ever taken a Z-Pak, that's what azithromycin does. Azithromycin blocks the exit tunnel on the 50S subunit of prokaryotic ribosomes. So this prevents this tRNA from exiting and the next tRNA from coming in. And if you don't do translation, you don't make proteins. You don't make proteins, you don't make enzymes. If you don't make enzymes, you can't live. And subsequently, these bacteria die. And antibiotics can act on any of the A, P, and E sites and several other sites. So that's why it's important to know the steps of translation as well as your exam.